Hello everybody and welcome back to a new video of IB Biology. In this video, we explore a new unit, Unit 1.3, where we explore the structure of biological membranes that make them fluid and dynamic. This is Chapter 1, Cell Biology. So, as always, I recommend you to download the IB subject guide, which gives you a great idea as to what the chapter and the unit expects of you. It gives you an insight into the nature of science and TOK aspects of the chapter, the understanding and guidances which are important concepts that you need to be able to know in this chapter and apply across different chapters or even different subjects, and applications and skills where you may need to know how to prove certain theories, how to apply certain theories, how to calculate or draw certain models. So let's move on. Phospholipids. This is an important concept that will be seen throughout this chapter as well as other chapters, so do take note. Phospholipids are the principal components of cell membranes. They are amphipathic molecules, which mean that they have both hydrophilic and hydrophobic regions. What these are, we will explore in the video later. The common basic structure of phospholipids is one phosphate group, one glycerol molecule, and two fatty acid tails, and this structure can be seen on the right hand side of the screen where it's drawn. Now it's important to know how to draw a phospholipid because it has come out in previous and past year exam papers as essay questions or structured questions where they've asked you to draw the phospholipid bilayer or the structure of phospholipids. So do take note and do memorize this drawing and understand what it's trying to say as well. Now, membrane proteins. Phospholipid bilayers are embedded with proteins which may either be permanently or temporarily attached to the membrane. There are two kind of proteins that you can find in phospholipid bilayers, namely integral proteins as well as peripheral proteins. So, integral proteins are those that are permanently attached to the membrane and are typically transmembrane. That means they cut across the entire bilayer. You will see them from the start to the end. Now, peripheral proteins, on the other hand, are temporarily attached by non covalent interactions and they associate with only one side of the membrane. That means they are monotopic, which means that either you'll find them on the top or on the bottom side of the membrane, but you uh, won't find them crossing the entire membrane or cutting through the entire membrane. So, the structure of membrane proteins, because they are proteins, the basic unit of proteins are amino acids. So, amino acids of membrane proteins are localized according to polarity. What does polarity mean? So, you have non-polar and polar. Non-polar means they're hydrophobic, polar means they're hydrophilic. Hydrophobic means fearing water. That means it's scared of water, so it likes to stay away from water. It doesn't interact with water. Polar, hydrophilic. Philic means you like it, so that means they like water, so they love to interact with water. So non-polar amino acids associate directly with the lipid bilayer. Lipids, as you know, fats are also hydrophobic. So example, if you put oil into water, the oil will float on top of the water. So the oil doesn't mix with the water and doesn't interact with it as well. That's why. Polar amino acids are located internally and face aqueous solutions. Aqueous solutions have a component of water, and as we know, polar, that means hydrophilic, they love water, so they love to stay around water. That's why they face aqueous solutions. Transmembrane proteins typically adopt one of two tertiary structures, single helices or beta barrels, or uh, commonly known as channel proteins as well. So these are how your integral proteins look like, as I mentioned, and peripheral proteins as well. Your integral proteins, there are a few kinds. You have an alpha helix structure, you have a helical bundle, you have a beta barrel, and all of these have different functions. So alpha helix sort of structures would probably be for receptors on the membrane proteins. Helical bundles you can see as in the form of enzymes, transporters, also receptors. Beta barrels can be seen in the form of transporters, which are also known as your channel proteins, which we will explore later in the chapter. Peripheral proteins, you can find them in the form of enzymes, anchorages, transporters, and many more. So functions of membrane proteins, because they are on the cell, obviously they have a very important role to play. So one, they are junctions. They serve to connect and join two cells together. Enzymes, they can be used to fix enzymes fixed to membranes, and they localize metabolic pathways. This is metabolic pathways is something we will explore in chapter 2. And enzymes, as we know, are biological catalysts. They are extremely important to the human body and many living things on earth. So the enzymes will interact with the membrane proteins in order to localize enzymatic reactions. Transport, obviously, as you know, diffusion, osmosis, active transport, all these happen across the membrane. And so your cell membrane, they regulate the inflow and outflow of molecules in and out of the cell. So transport again. Recognition may function as markers for cellular identification. There are so many cells in your body, and because you have an immune system, the body needs to know which cell belongs to the body and which is a foreign particle. So your membranes can be used as a recognition method. Anchorage, they can be used as attachment points for cytoskeleton or extracellular matrix. And transduction, which functions as receptors for peptide hormones. Hormones, we will explore later in chapter 6 as well. So it is important to regulate the degree of fluidity, because as you mentioned, the membranes are fluid and dynamic. So they need to be fluid enough in order for the cell to move. But if they are too fluid, the membrane could not effectively restrict the movement of substances across itself, right? If it's too fluid, substances will just go in and out, and then the whole purpose of your membrane is just defeated. Membranes need to be fluid enough such that the required substances can move across, and those substances that are not allowed to or should not move across can be stopped right at the start. So the phospholipid bilayer, as we know it, the structure, it consists of one polar head, polar hydrophilic, and two polar tails, which are hydrophobic. That means they hate water. So the hydrophilic part is composed of glycerol and a phosphate molecule, and the hydrophobic tails are composed of fatty acids. So fats, once again, remember, they don't exactly like water, so they're hydrophobic, and you have two of them. So two non-polar hydrophobic fatty acid tails. uh, Phospholipids spontaneously arrange themselves into a bilayer. So the hydrophobic tail will face inwards, so they are shielded from the surrounding fluids and aqueous solutions. And the hydrophilic head, on the other hand, will associate with the cytosolic and extracellular fluids respectively. So if we see over here, you can see that these are the hydrophilic heads. So because there's a bilayer, that means there are two layers by, so two. So the heads will face on the outside because they love water. So on the inside over here is your cytoplasm. So your cytosolic fluid is here. Here is extracellular, so your extracellular fluid, so outside the cell. So any fluid or any aqueous solution that the membrane has to face, it will be faced by your hydrophilic heads. Now your hydrophobic tails, because they hate water so much, they will face each other and inward. So there's no aqueous solution there. They won't come into contact with water, so they're safe. Now, properties of the phospholipid bilayer. The bilayer is held together by weak hydrophobic interaction between the tails, as it makes sense because the tails itself are hydrophobic, so there will be weak hydrophobic interactions as well. The hydrophilic and hydrophobic layers restrict the passage of many substances, and individual phospholipids can move within the bilayer, allowing membrane fluidity and flexibility. And this fluidity actually allows for spontaneous breaking and reforming of the membrane, as we will learn later in endocytosis and exocytosis in later units of the same chapter. So once again, you are required to know how to draw the plasma membrane. You are required to know how to label each of these proteins. You are supposed to know their functions, as well as how they contribute towards the fluidity and the dynamics of the phospholipid by layer membrane. So do memorize this drawing and do learn the various components that are part of this membrane itself. This also comes out as an essay question or as structured questions, or even an MCQ many of the times, where they may ask you to maybe identify a certain protein or certain component, or ask you the functions, or even if they ask you about the phospholipid bilayer in an essay question, just drawing this diagram itself sometimes can lead you to getting 5 marks out of 8 marks, for example. So it's very, very important that you know this drawing. It's part of the syllabus guide as well if you do have a look. So now, moving on to learning the different components of the phospholipid bilayer itself, its functions and characteristics as well. So glycoproteins, proteins which have an oligosaccharide, which means there are few sugar molecules attached, so it's a whole chain, and is important for cell recognition by the immune system and as hormone receptors. So as we knew, as we studied in the previous slides, the function of a membrane can be used to serve as a recognition mechanism. So this is where your glycoproteins come in. So once again, glycoprotein looks something like this, the structure over here. So it's used for recognition and receptors as well. Now, proteins. As I mentioned, you have two kind of proteins: integral and peripheral. So, integral, as you can see, they will run throughout the entire membrane. So, channel proteins and carrier proteins are two kinds of integral proteins that you see in a bilayer. So, as you can see, they are transmembrane. They run throughout the entire membrane. And then your peripheral protein, which is monotopic, that means it only attaches itself temporarily sometimes to only one surface or one part of the bilayer, so it's just on one of the sides. All right, so integral proteins, they are permanently embedded, that means they're just going to be there. They are polytopic, that means they go through many surfaces. And sometimes you also have integral proteins, they may be monotopic, but mostly are polytopic in nature. Now, peripheral proteins, they usually have a temporary association with the membrane, and they can be monotopic or attached to the surface. So cholesterol is also very important part. As you can see in my cursor over here, cholesterol is here. It's a very important part of the membrane. Now, cholesterol itself is a component of animal cell membranes, where it functions to maintain the integrity and mechanical stability of the membrane. So it is absent in plant cells because, as you know, plant cells, they have a cell wall which is made of cellulose, so they do not need this extra cholesterol in their cell membranes to make the cell even more rigid. It doesn't help. So cholesterol, once again, is an amphipathic molecule, which means it has both hydrophilic and hydrophobic regions. Now, the functions of cholesterol itself, it interacts with the fatty acid tails. As you can see in the drawing over here, where it's drawn, it is strategically placed very closely to the fatty acid hydrophobic tails. So it interacts with them in order to moderate the properties of the membranes, and this is very important. One, it reduces fluidity. So, like we mentioned, it's important to regulate the fluidity of the membrane itself. It cannot be too fluid and it cannot be too rigid as well. So it ensures a certain level of maintained fluidity of the membrane. So it functions to immobilize the outer surface of the membrane, so it's not moving too much. It reduces permeability because the cell membrane, at the end of the day, is supposed to regulate substances moving in and out of the cell. So it reduces permeability to ensure that it can function as so. It prevents crystallization of the membrane, which functions to separate the phospholipid tails to prevent crystallization of the membrane. And it secures the peripheral proteins because, as we know, they are temporarily attached and they are only monotopic. So by forming high density lipid rafts, they are capable of anchoring the protein to ensure that the protein does not just float away into the extracellular space or into the cytosolic fluid. Now, membrane models, as you will learn in this chapter, there were two models that are extensively discussed in this chapter: the Davison and Danielle model, which is also known as the lipoprotein sandwich model, and the Singer Nicholson model, which is also known as the fluid mosaic model. So, the initial model that was proposed and accepted was the Davison Danielle model, which attempted to describe the position of proteins within the bilayer. So initially, when they did have a look, they thought that the bilayer or the membrane was actually trilamina, which meant that they thought it was three layers: two outer dark layers and one lighter inner layer, as you can see in this micrograph on the right hand side where my cursor is pointing. So Davison and Daniele proposed that the model whereby two layers of the protein were flanked with a central phospholipid bilayer. The model itself was described to be a lipoprotein sandwich, as such that the lipid layer was sandwiched between the two protein layers. So on top over here, these two dark lines were the protein layers, and in the middle smack was the lipid layer. The dark segments on under the electron microscope were then identified wrongly, as we know, because there was a falsification of the Davison Danielle model. Now, this falls under TOK as well and nature of science. So, you are to know the falsification as to why the model was falsified, on what grounds it was falsified, and what was used to falsify the model. So, you are needed to know this because it comes out many times and it's a very popular essay, MCQ, and even structured question in exam papers. So the falsification of them, the number of problems with the lipoprotein model were too many. So one, they assumed that there was uniform thickness. They assumed that all the membranes, which meant that they assumed all the membranes were of uniform thickness and would have constant lipid protein ratio. The surfaces were also assumed to be symmetrical internally and externally, and the permeability was not accounted for the membrane, and the temperature at which the membrane solidified did not correlate with the expected under those of the proposed model. So these were the problems that were faced out and flagged out of the Davison-Danieli model. And what was used to flag out these problems were these methods. So firstly, they use fluorescent antibody tagging as well as freeze fracturing. So, as we know, membrane proteins are insoluble and varied in size. As we know, there's a phospholipid head, there are integral proteins, there are peripheral proteins. So, how can you expect the layer to be uniform and to be symmetrical when you have proteins bouncing in and out, and you have glycoproteins and you have cholesterol? So, there's no way that you would have a uniform and continuous layer on the outer surface, as mentioned and explained by Davison and Daniele in their model. So, firstly, fluorescent antibody tagging was used, which showed that the membrane proteins were actually mobile and not fixed in place. The proteins from two different cells were tagged with red and green fluorescent markers, respectively. So when the two cells fused, the markers became mixed throughout the membrane of the fused cell, and this then hence demonstrated that the membrane proteins could move and were not actually a static layer as per the Davison-Danieli model. Now, freeze fracturing was another method which was used to split open the membrane, and that revealed an irregular and rough surface of the membrane. The rough surfaces were then interpreted as being transmembrane proteins, as you know, the integral proteins, demonstrating that proteins were actually not solely localized to the outside of the membrane, but some of them actually uh, were transmembrane, so they cut throughout the entire membrane and throughout the bilayer as well, which is when the Singer-Nicholson model came in after the Davison-Danieli model was falsified. They got to know that no, it is not a trilamina. That means there were no three layers; there were actually two layers, and the many false. And the many problems that they found in the Davison-Danieli model were then proved, and hence we had a new model that came up, which was the Singer-Nicholson model, also known as the fluid mosaic model. So, in light of these limitations that were seen in the Davison-Danieli model, a new model was proposed by Seymour Singer and Gareth Nicholson. So these are actually four important names that you need to know: Davison, Daniele, Singer, and Nicholson. And these people actually come out a lot in MCQs when they ask you the TOK aspect of this chapter. So, according to this model, according to the fluid mosaic model or the Singer Nicholson model, proteins were embedded within the lipid bilayer rather than existing as separate layers. And this model got to know as a fluid mosaic model, and this is the model that is preferred by scientists today, and this is the model that we refer to in our drawings and also study, and is what is widely accepted. So, membrane fluidity. So, once again, cell membranes were fluid. This was proved. And the phospholipid structures, as we know, phospholipids, they vary in length. They have relative saturation as well. So, the shorter fatty acid tails will increase fluidity as they are less viscous and hence more susceptible to changes in kinetic energy. And the lipid chains with double bonds, unsaturated fatty acids, they have kinked hydrocarbon tails so that are harder to pack together, so that results in the model being fluid. So cholesterol, as mentioned in the previous slides, they act as a bidirectional regulator of membrane fluidity. They help to regulate the fluidity. At higher temperatures, it actually stabilizes the membrane and raises the melting point to ensure that the membrane does not melt and dissociate. And at low temperatures, it intercalates between the phospholipids, preventing clustering. So when the temperatures are low, it prevents the membrane from collapsing and becoming too rigid. So that's how it functions. It's very, very important. Cholesterol is extremely important in the membrane itself. And that's it. That's the last slide of unit 1.3, where we explored the bilayer membrane and its fluidity and dynamics. Thank you so much for watching this video, and I hope you were able to learn unit 1.3. I will be back with more videos to explore the rest of the units of chapter 1, cell biology. Thank you.